Hello everyone, my name is Jeannette Bouchard and I am an infectious diseases pharmacist at WakeMed Health and Hospital System. In this presentation, I will be reviewing the pertinent drug information regarding the SARS-CoV-2 viral vector vaccines. In this video, we will be discussing the SARS-CoV-2 vaccines utilizing viral vector technology. As of December 26, 2020, there are 39 vaccines utilizing viral vector technology in preclinical and 16 in clinical development. The vaccines seen on the slide are the 4 that have current phase 3 trials underway. These will be the focus of the current video and the future updates. AZD1222 from AstraZeneca will be presented here. This vaccine candidate is currently being studied as a 2-dose regimen 28 days apart. It should be stored at 2 to 8 degrees Celsius and is currently projected at a cost of $3 to $4 per dose. Viral vector vaccines rely on another virus that is engineered to express the desired antigen. In SARS-CoV-2's case, this would be the spike protein. Depending on the virus type, it is either designed to be non-replicating or replicating. Non-replicating vector vaccines are not able to make new viral particles and only produce antigen, whereas replicating vector vaccines produce new particles in the cell it infects, which will then go on to infect new cells and make antigen. The current SARS-CoV-2 vaccines use non-replicating vector vaccine technology. Research on these vectors has been conducted since the 1980s. Numerous viruses have been utilized as the basis for constructing these vaccines, including adenoviruses, parvoviruses, and togaviruses. Adenovirus vectors are one of the most utilized vectors and is the vector of choice in some SARS-CoV-2 vaccine candidates. Adenovirus vectors are ideal because they are able to stably support the expression of most target antigens as well as multi-pathogen vaccines. These vectors can also be produced in systems that support a high viral yield at low production costs, a trait that is important when creating a vaccine during a pandemic. Viral vector vaccines generally produce a strong immune response and do not need additional adjuvants like protein subunit vaccines would. When tested in combination with immune-stimulating components, no added immunity was seen. Adenovirus vectors specifically are able to produce a potent CD8 T cell and antibody response. One downside to adenovirus vectors is the pre-existing immunity to the virus in humans that may inactivate the viral vector. This is why we seek vaccine candidates with vectors, such as the chimpanzee adenovirus, to ensure that there is limited to no pre-existing immunity to these vectors. First, we will review the preclinical phase 1 and phase 2 data for AstraZeneca's AZD1222. AZD1222, formerly ChAdOx1 nCoV-19, is an adenovirus vectored vaccine that encodes a codon-optimized full-length spike protein of SARS-CoV-2. The design for this vaccine was based on a previous vaccine platform used for MERS-CoV, which utilized a chimpanzee adenovector vaccine encoding the spike protein for MERS. This vaccine showed protection in non-human primate models against MERS-CoV-induced disease. Non-clinical data for AZD1222 is summarized on this slide. This information is in preprint format and has not yet been peer-reviewed. In this study, vaccinated mice showcased IgG titers against spike protein subunits and neutralizing antibodies in all vaccinated mice compared to none of the mice in the control group. A strong Th1 T cell type response was noted post-vaccination in mice. In the non-human primate evaluation, 6 macaques were vaccinated with 2.5 times 10 to the 10th AZD1222 virus particles each. This is half of the currently administered human dose in some cases. IgG antibodies against spike protein were present at 14 days post-vaccination, and neutralizing antibodies were detectable by day 28. Upon re-challenge with SARS-CoV-2 to upper and lower respiratory tracts, the control animals had viral RNA detected in BAL fluid on all days. In vaccinated animals, 2 out of the 6 had detectable viral RNA in BAL. Viral RNA was detected in nasal swabs in all animals on any day, showing no difference between vaccinated and controlled animals. These results are depicted on the image here. The red circles represent the vaccinated animals, and the blue squares represent the control animals. The immune response noted in the study was not predominantly a Th2 T cell response in both mice and non-human primates. This promising animal data led to the continuation of Phase 1 and Phase 2 dosing and safety trials. The study breakdown for AZD1222 is represented here. Currently, there are 4 trials underway. COV-1 is a single-blind Phase 1-2 clinical trial out of the UK. This will be discussed in more detail over the next few slides. It was originally planned as a single-dose trial, however, only 88 patients still remain in the single-dose group of the study. COV-2, which is a single-blind Phase 2-3 study out of the UK, is evaluating 2 separate dosing groups for AZD1222. A low dose, which has a prime defined as 2.2 times 10 to the 10th viral particles, with a standard dose booster defined as 5 times 10 to the 10th viral particles, or 2 standard doses for the prime and booster doses. Older adults in this study only received the standard 2-dose regimen. COV-3 was a single-blind Phase 3 study out of Brazil that is evaluating a 2 standard dose regimen given up to 12 weeks apart with a target revaccination of 4 weeks. And COV-5 is a double-blind Phase 1-2 study out of South Africa that will be evaluating 2 standard doses 4 weeks apart. This study will eventually have an immunogenicity cohort of those living with HIV. These results are not available at the time of this video. The published Phase 1-2 study, COV-1, was a randomized single-blind controlled trial that compared AZD1222 to the meningococcal vaccine. The meningococcal vaccine was used as a comparator vaccine to maintain blinding of participants who experienced local or systemic reactions. This study included healthy adults ages 18 to 55 years who were separated by 4 different groups. Of these groups, Group 3 consisted of 10 patients who received a booster of the vaccine 28 days after the first dose. The dose for the vaccine was 5 times 10 to the 10th viral particles, which equates to the standard dose that was described in the previous slide. The co-primary outcomes were number of symptomatic virologically confirmed COVID-19 and safety of the vaccine measured by the occurrence of serious adverse events. Secondary outcomes included safety, reactogenicity, and immunogenicity of AZD1222, and efficacy against hospitalized COVID-19, death, and seroconversion against non-spike proteins. A total of 1,077 participants were enrolled in COV-1, with 543 assigned to AZD1222 and 534 assigned to control. In the AZD1222 group, antibodies against spike protein peaked by day 28 and persisted until day 56 in the single-dose group. The booster dose group saw a similar antibody effect, but an increased median at day 56, 119 ELISA units in the single-dose group versus 639 ELISA units in the booster. Neutralizing antibodies were exhibited in all participants after a booster dose. At baseline, there were participants with high levels of neutralizing antibodies, and when vaccinated, these antibodies were boosted, showing a potential benefit in patients who may have been previously exposed to the virus. Regarding safety findings in COV-1, AZD1222 saw higher local and systemic reactions when compared to the control group. However, some of the local reactions, such as pain after vaccination, were mitigated by the use of paracetamol. The most common systemic reactions noted were fatigue and headache, which were reported in 71% and 61% respectively. Local and systemic reactions were highest 1 day after vaccination. Other common systemic reactions reported were muscle ache, malaise, chills, and fever. In the group of prime boost participants, systemic and local reactions appeared to be lower following the second dose. Given the small number of participants in this group, it is difficult to decide if this profile will continue with larger participant groups. Unsolicited adverse events that were considered possibly, probably, or definitely related to the treatment arm were generally mild to moderate in nature. Of note, neutropenia was observed in 25 of 54 participants in the treatment group compared to 3 of 44 in the control group. Overall, AZD1222 was safe and tolerated and had a better tolerability with the use of paracetamol prophylactically. Humoral responses peaked by day 28 and cellular responses appeared by day 14. These responses were more potent in the prime booster group, and this led to further clinical data needed regarding the prime booster regimen rather than the single dose. Next, we will discuss the pivotal efficacy trial for AZD1222. Boise and colleagues recently published an analysis from the 4 ongoing blinded randomized controlled trials described earlier in this video. Participants were ages 18 and older and were randomly assigned in a 1-to-1 ratio to receive either AZD1222 or control, which depending on the study site could have been saline or the meningococcal vaccine previously described in COV-1. There was a small subset in COV-2 who received a half dose as their first dose of the 2-dose regimen. However, the rest of the participants in the study received 2 standard doses of vaccine that were targeted towards a dose of 4 weeks apart. However, there were large intervals when the dose could not be given. For COV-2, for instance, 4 to 6 weeks was sufficient, and for COV-3, up to 12 weeks was allowed. 53.2% of participants in COV-2 in the low-dose standard dose group received a second dose at least 12 weeks after the first, and only 0.8% received a second dose within 8 weeks of the first. The primary objective was to evaluate the efficacy of AZD1222 against symptomatic COVID-19. This was defined as positive serology in at least one qualifying symptom. COV-5 was the only study that addressed events more than 14 days after booster as part of their primary outcome. However, the primary outcome in the pooled analyses specifically addressed infection more than 14 days after the second dose of vaccine. Secondary efficacy endpoints include severe COVID-19 disease, hospital admission associated with COVID-19, ICU admission associated with COVID-19, and deaths associated with COVID-19. Antibodies against SARS-CoV-2 non-spike proteins were also assessed in the secondary efficacy endpoints. There was a total of 23,848 participants recruited and vaccinated across all 4 studies: 1,077 in COV-1, 10,673 in COV-2, 10,002 in COV-3, and 2,096 in COV-5. Baseline demographics were broken down based on study and are seen here on this slide. A majority of participants were female and white. There was also a high percentage of health and social care workers included in all trials in the pooled analysis. Regarding comorbidity breakdown, around 10% of participants in each study had cardiovascular disease and around 10% had respiratory disease, with very few having diabetes, between 1 and 3% depending on the study. Notably, COV-2, which is the only study to include a low-dose standard dose regimen, only enrolled participants between the ages of 18 and 55 years old, and overall, across all studies, there was a limited number of participants enrolled above the age of 56 and even less so above the age of 70. Of the approximate 11,000 participants included in the primary efficacy analyses, there were 30 cases in the vaccine arm and 101 cases in the control arm, resulting in a vaccine efficacy of 70.4%. For participants who had any serologic COVID-19 test result, there were 68 cases in the vaccine arm and 153 in the control arm, which equates to a vaccine efficacy of 55.7%. Additionally, certain studies were designed to assess asymptomatic disease in participants. Of those that were included in this analysis, there were 29 cases in the vaccine arm and 40 in the control arm, resulting in a vaccine efficacy of 27.3%. For the secondary efficacy endpoint of hospitalized patients, there were 10 participants hospitalized more than 21 days after their first dose of vaccine. 2 of these were considered severe COVID-19 cases, and there was 1 fatal case. All cases that resulted in hospitalization or severe COVID-19 and death were in the control group. Results from the subgroup analyses provided in the pooled analysis are shown here. When broken down between the low-dose standard dose and the 2 standard dose regimens, the vaccine efficacy saw a separation, with the low-dose regimen having 90% vaccine efficacy and the standard dose regimen resulting in 59.3% vaccine efficacy. When further evaluated by dosing interval between these 2 dosing regimens, the low-dose regimen still showed a higher efficacy of 90% when the dosing interval was greater than 8 weeks. For all standard dosing regimens in the UK and Brazil, when grouped by redosing interval, there was a numerically lower vaccine efficacy in the redosed group less than 6 weeks. However, the confidence intervals associated with these subgroup analyses are large, and more data is likely needed to elucidate the true effect of these dosing intervals and regimens for AZD1222. For key safety outcomes noted in the pooled analyses, local and systemic reactogenicity appeared to be lower and less intense in older adults, the low-dose regimen, and after the second dose. Serious adverse events and adverse events of special interest were balanced between vaccine and control arms in the analyses. Of the 175 events reported, 3 were considered possibly related to either the treatment or the control group. One of these events was a case of hemolytic anemia in the control group. The second was transverse myelitis in the vaccine group, and the third was a fever of greater than 40 degrees Celsius that was reported 2 days following vaccination. It is not known which group this participant fell into, but they received the second dose without a subsequent reaction. There were 2 additional cases of transverse myelitis noted during these trials that led to a temporary pause of the clinical trials for AZD1222. These 2 were later deemed to be unrelated to the vaccine by an independent committee of neurologic experts. Regarding deaths, so far in the pooled analyses, there were 4 non-COVID related deaths across all studies, 3 in the control group and 1 in the vaccine. All were considered unrelated to the study. In summary, based on current data for both preclinical and clinical trial data, AZD1222 is safe and effective against COVID-19 with 70.4% vaccine efficacy. Serious adverse events were low and consistent between treatment and placebo arms. And contrary to the mRNA vaccine candidates described in a different video, local and systemic reactions following the second dose were less intense. Although they were less intense, it is still important to note that the systemic side effects were more frequent in the treatment arm, and this should be taken into consideration when vaccinating groups of people such as healthcare workers. Systemic symptoms can overlap with many of the currently utilized symptom screening tools for workplaces, and encouraging vaccination at the end of the work week may be appropriate to avoid overlap. This analysis pooled studies from a variety of global sites, including Brazil, South Africa, and the UK. Additionally, it included a large proportion of participants who were at a particularly high risk of contracting disease based on their occupations. Although limited, there were some data on AZD1222's effect on asymptomatic disease. As these trials continue, this will be an important factor to consider when evaluating the differences between the available vaccines on the market. All of these factors, perhaps one of the most important aspects of this vaccine is the cost per dose relative to other vaccines, which is $3 to $4 per dose compared to $20 of some others. It is also easy to store as it only requires refrigeration. In terms of what is currently missing from data, the biggest data point that is missing is the optimal dosing regimen and interval. Based on the pooled analysis, the accidental lower dosing regimen of AZD1222 seems promising in comparison to the efficacy outcomes for the standard dose regimen. Although, this must be interpreted cautiously, as the lower dosing regimen was only given to a limited number of participants in one study, and it did not include older participants. Other major gaps for the general population are the long-term safety and efficacy data, as well as the vaccine's efficacy against severe disease. There were too few cases of severe disease to be conclusive about any kind of overwhelming efficacy for prevention of severe disease or hospitalization. Further data will be needed to address these issues, and until then, continued public health reminders, such as mask wearing and social distancing, is strongly encouraged. Further data is also needed in specifically excluded populations such as pregnancy, breastfeeding, and immunocompromised. There are currently no data available to speak to safety and efficacy outcomes in these populations. As more data from clinical trials and patients receiving the vaccine become available, we will hopefully have more information to speak to these populations. Specific resources to learn more about vaccine recommendations and frequently asked questions are listed here on this slide. The CDC provides excellent guidance for both healthcare providers and patients receiving vaccine, as well as separate information for the EUA-approved vaccines available. Thank you very much for watching. I hope this was a helpful summary of the SARS-CoV-2 viral vector vaccines. Again, this video will continue to be updated as information becomes available. If you have any questions regarding the material presented in this video, please reach out.